Hello friends, today we will discuss about the most important topics of molecular biology, and just I will highlight the most important topic for your upcoming NEET PG and INICT examination. So the first is the nucleotide and you have to remember just few topics regarding nucleotides, like both the both the anabolism and catabolism of the purine and pyrimidine nucleotides. Okay, this is very important. If you have time you can read the all, but it is the most important of the anabolism and catabolism of both purines and pyrimidine. But you have to already also you need to know the structure and where the nitrogen is come from, where the carbon is come from. From the purine you have to remember the basic point that purine is, if it is a catabolism, it is produce the uric acid, and in case of pyrimidine it is produce ammonia, it is beta alanine, beta amino isobutyrate, and CO2. Okay, you also need to remember about the associated disease for the purine and pyrimidine, and you know betterly that in case of purine it is the Lesch-Nyhan syndrome. In case of pyrimidine metabolism it is orotic aciduria, and in case of purine partial AGPRTS deficiency it is Kelly syndrome. Kelly-Sigmiller syndrome. Next, second, you have to remember the important topic, that is the replication. If you have very less time, the most important topic is the three unwinding proteins of the for replication, and you know it is helicase and topoisomerase and single stranded binding proteins. Okay, so you have to remember this, and you have also need to remember the DNA polymerases, that is DNA polymerase 1, DNA polymerase 2 and DNA polymerase 3. Okay, and you know about the DNA polymerase 1, which is responsible for formation of clinofragment. So it is also very important, clinofragment, that is the N terminal to C terminal, and N terminal there is a 5' to 3' exonuclease, then 3' to 5' exonuclease, then 5' to 3' polymerase. Okay, and then वो पहले वाला ये होता है. Next is very important, that is the transcription, and you have to know about the RNA polymerases. Like in case of eukaryote it is 1, 2, 3 also. Okay, or in case of prokaryote it is a hollow engine in which sigma, you know about the sigma is responsible for binding of the site for attachment. Next, you have to also know about the strand of synthesis, about the antisense and non-coding determination. And I am just showing you how it is determined about the antisense or non-coding in case of new strand of RNA. So you have to know it. It is very important, and I am following this question in many GTs and also in mock tests. So this is the two strands of DNA. One is 3' to 5'. This is the 5' to 3', and this is the newly synthesized RNA 5' to 3'. Suppose in the upper strand there is a in the upper 3' to 5' strand there is A, T, G, C, A and T. So in the 5' to 3' strand it will be T, A, C, G, T, A, and you know in RNA there is no T, so it is U, A, C, G U A. So you look for, there is a very similarity, that is A C G A. So we can consider this 5' to 3' of the previous DNA strand as it is like that non-template strand, also coding strand, sense strand, positive strand. So it is very important for this upcoming NEET and INICT examination. Okay. Fourth, you have to know about the lac operand. Very important topic for this year NEET and INICT, and very much chance of probability of exam in this year. Fifth, you have to know about genetic codes, characteristic like the degeneracy, universality, okay, and commalase, okay, unambiguosity. You have to know, and I think you have to know this. So this is very important for your single one-liner questions. Sixth is you have to know about the B-A-Z DNA difference. Okay, this is also a last-minute topic for your upcoming exam. And you have to also know the types of restriction in a nuclease. You know about the type 1, type 2, type 3, type 4, in which type 1 is away from the recognition site, type 2 is near the recognition site, type 3 is parendomyxin, type 4 is obviously methylated DNA sequences. Okay, you know about this. And obviously you have to also know about the siRNA and miRNA, and you know about the two questions already had come in INICT July session 2021. Two questions on this. One is knockdown question, another is where it is bound at the 3' UTR region. Okay, so it is also very important topic. Okay, and also there is some topics like you also know about, that is the PCR, in which Mg2 plus and buffer is needed. This Mg2 plus question I have found in some GTs. Also you have to know about RFLP, restriction fragment length polymorphism, also about microarray. This is the very small topic. This is also overlapping with genetics topic. I am just doing chromatin immunoprecipitation. Next is fish. Okay, and the remaining topic I will genetics, whatever left I will discuss in genetics. So okay, thank you friends. If you like this video, please